right, folks, it's 10:02. We'll go ahead and get started this morning. Very happy New Year to everybody. Welcome to the South Atlantic LCC's Third Thursday Web Forum. This is the January edition, and as usual, I'm Hillary Morris, and I work on Blueprint User Support and Communications for your cooperative. So this is a monthly event. We host this web forum once a month featuring a presentation on current and ongoing work throughout the South Atlantic LCC. It's every third Thursday of the month at 10 a.m., and it's a great chance to ask questions, provide feedback that will help guide the future of your cooperative. So every month we basically have the same agenda. I'll introduce our presenter who will speak for 30 to 40 minutes. There'll be plenty of time for questions after the presentation, and then we'll spend the rest of the webinar on updates from your cooperative staff, followed by an open discussion. So before I hand things off to Josh, a few logistics. I do mute everyone during the presentation just to minimize background noise. So if you have any questions, you can either type them directly into the chat box and I'll monitor that, or you can hold them until after the presentation. So if you're using the chat box and you want everyone to be able to read the question, just remember to send the note to everyone. And to speak at any time, all you need to do is press *6 to unmute yourself. So before I put us in silent mode, does anyone want to speak up with questions, comments, or concerns?
The conference is now in silent mode.
All right. So presenting today, we have Josh Epstein, who's a graduate research assistant at the University of Florida in wildlife ecology and conservation. So he'll be giving us an update on the results of a project that was funded by the South Atlantic LCC to test the freshwater aquatic and as well as the waterscapes indicators. So I'm going to pass presenter power over to you to get us started. Josh?
Thanks, Hillary. Can you hear me?
I can hear you great.
Okay, perfect. Okay, well, hi everyone. Thanks for attending my presentation this morning. As Hillary stated, I'm Josh Epstein. I'm currently a master's student at the University of Florida. And for my project, I've been working with the SALCC to test some of their indicators of aquatic ecosystem integrity. Today I'm going to talk to you about my work on the functional diversity of South Atlantic LCC fish communities.
Hey Josh, we can't see your slides. If you wouldn't mind sharing your application again.
Sure, sorry.
No worries. Not that you necessarily need illustrations to introduce yourself, but
oh, you got to drag the little ball. I think. Go back to the little ball.
Where?
You've got the POWA, so just go into Share Application PowerPoint.
Okay, I got to go back there.
Yeah.
Gotcha.
That ball.
Now we're cooking.
Okay, perfect.
Thank you. All right, it's all yours.
Okay. Except you can probably see this.
Yeah, I can still see your presenter view. Thanks for bearing with us, everyone. Beautiful. Okay. We're in business. Take it away.
Sorry about that. All right. So just to give you a quick outline of what I'll be discussing with you, I'm going to begin by discussing functional diversity, what it is, why it's important, and then I'll discuss how we measure functional diversity. I'll then get into our specific approach for analyzing functional diversity across the SALCC and for relating levels of functional diversity with some of our indicators. And as most of you already know, something that the SALCC is interested in is understanding how environmental variables are related to indicators of biodiversity at large scales. And we're often interested in analyzing the relationships between a large suite of environmental or landscape or watershed variables with biodiversity indicators. And we often look at species richness in this context. And we often overlook other important measures of diversity, which give us additional and often more information than species richness alone does. And for this reason, for our study, and in an attempt to test some of the SALCC indicators, we explore relationships between these indicators and freshwater fish functional diversity within the SALCC region. But what exactly is functional diversity? Well, functional diversity represents the overall diversity of traits in a system, and functional diversity can be correlated with species richness, but often values of functional diversity are different than those of species richness. So species richness is important. It doesn't often disclose a lot of information about what's actually going on in the system or about the functioning of a system, and it really just takes that first step in telling us who's there and who's not. So on this slide, you can see an example of the potential differences in functional diversity with similar species richness, and we're looking at two hypothetical lakes here. So if you start by looking at Lake 1, you can see that there are 3 species of sunfish, and they're all from the same genus. They're all the same size. They all eat the same things. They all have the same habitat preferences, and therefore there's a lot of trait redundancy in this system. So conceptually, we would say that there's low functional diversity here. And this is an example of what we might see in degraded systems with poor conditions. A lot of similar generalist species that can tolerate these poor conditions, such as anoxia or a surplus supply of nutrients. But on the other hand, if you take a look at Lake 2, we have 3 different species again. So we have the same species richness, but these species are all from different genera, and they're all different sizes. They all eat different things. They all have different habitat preferences, and therefore there's a lot of difference in traits represented. So here we would say that we have high functional diversity. So even though we have the same species richness, we have a large difference in the diverse traits expressed in these lakes, and this often can reveal important information about a system. And because it deals with traits, functional diversity is really important in influencing things like ecosystem dynamics, stability, productivity, nutrient balance, and other aspects of ecosystem functioning. And it therefore gives more information about what's going on in the system than species richness does alone. And it can also be directly related to ecosystem services because organismal traits are often those that provide for specific services. And examples of this could be things like the way that insect traits have an effect on pollination, or how bacterial traits affect the carbon cycle. And these things are directly tied to ecosystem services. And for these reasons, functional diversity, which in our study served as a proxy for ecosystem health, could potentially be used to measure or predict specific watershed or landscape indicators that the SALCC is interested in. So there's many different metrics that are used to measure functional diversity, and they usually utilize a species by species distance matrix, which is generated from a trait dendrogram, which is generated obviously from a list of traits that a group of species exhibit. And some of the most commonly used metrics for measuring functional diversity are the mean nearest neighbor metric, which measures the mean distance of each species' closest functional neighbor based on that dendrogram. Another metric is the pairwise functional distance metric, which measures the mean dissimilarity of trait values at each site. And the functional dispersion metric, which is what we decided to use in our study because it is unaffected by species richness. It's flexible in that it can handle any number and type of traits, and it's not strongly influenced by outliers. And what it does, functional dispersion measures the distance in multidimensional trait space to the centroid of all species in the community. And again, I'm going to use our 2 lakes from a couple of slides ago or from the last slide to exemplify the idea of functional dispersion. So you can see here we have 2 plots, and we have 1 plot for each lake. On the x-axis, we have maximum total length, which is one trait that we might be interested in analyzing. And on the y-axis, we have longevity, which is another trait that we might be interested in. And here we're just looking at these 2 traits just to keep things simple, but in reality, we often look at many different traits, which would simply be, which would simply be additional axes on these plots adding to their multidimensionality. And the species locations on the plots are their corresponding value for each of the 2 traits. So starting by looking at Lake 1's plot, if you recall, Lake 1 had low functional diversity. We have 3 sunfish, all from the same genus. And you can see that here, all species are close to the centroid, and therefore the average distance to the centroid is low. So that's why we see low functional diversity in Lake 1. If we take a look at Lake 2, which had high functional diversity, 3 different species from different genera, you can see that the three species are all a lot further away from the centroid than those in Lake 1, and therefore the average distance to the centroid is higher, and that's why we have high functional dispersion in Lake 2 compared to Lake 1. So our overall study objective was to relate environmental indicators and other environmental variables to freshwater fish functional diversity across the SALCC. And the idea here is that if specific indicators are significantly and highly correlated, then functional diversity could successfully be used to predict values of these indicators. And these are the ones that might be most important from a conservation standpoint because they're related to the diversity of traits in these systems. Okay, so as I'm sure you already know, the SALCC spans from southern Virginia down to northern Florida and slightly into eastern Alabama, and we also have portions of the Gulf included in the region. Here you can just see a breakdown of the HUC-8s within the SALCC, and HUC-8s are outlined in blue on the right, and the SALCC boundary is outlined in red. And we chose to include all HUC-8s within or touching this red boundary in our functional diversity analysis. So we ended up measuring functional diversity using the functional dispersion metric for 121 HUC-8s within the region. So what we did next was to determine the species present within each of these 121 HUC-8s. We used a couple of different databases. First, we used the Multi-State Aquatic Resource System, or MARIS database. And this database is an online resource that contains over 1 million fish sampling and water quality records for over 1,000 fish species compiled from State Fish and Wildlife Agency and other sampling programs. So we used that one. In addition, the data period for that, as you can see, is 1953 to 2012, and many different fish sampling efforts within that time period. We also looked at the USGS non-indigenous aquatic species database to capture some of our non-indigenous fishes. And this is an online resource and repository for spatially referenced biogeographic accounts of introduced aquatic species. And it's really important here to note that from these two databases utilize fish sampling efforts from many different studies, and that was one of the goals of this project. It was kind of to use these larger databases to get a regional identification of the species that are present. But as we'll see, there are both positive and benefits to using this approach. And for this analysis, we assume that if a fish species was found at any time within a HUC-8, we considered it a part of the community. And we know that this is a coarse measure of community composition, but as I kind of touched on when working with data from many sampling efforts, this is often the best we have to work with. But a potential option to deal with this that we have yet to explore is to run a sensitivity analysis to potentially remove some low occurrence species and see if our results are impacted. So once we had species presence absence for each HUC-8, we determined the fish trophic, reproductive, and habitat traits from primary and secondary literature, the Virginia Tech Fish Traits Database, and expert knowledge. And these tables you see here at the bottom show the trophic, reproductive, and habitat trait topics that we looked at for our analysis. So in terms of trophic traits, we were interested in both what these fish eat and where in terms of water column position they eat it. In terms of reproductive traits, we were interested in egg guarding behavior, spawning behavior, and spawning location. And for habitat traits, we were interested in substrate type, water body type, spawning movement, aka anadromous or diadromous fish, elevation, and current type. After taking all of these into account, we analyzed 11 specific trophic traits, 19 reproductive traits, and 25 habitat traits. So what we did, we basically broke these broader trait categories down into specific traits, and these were the amounts that we got. And if a species had a given trait, the species was marked as a 1 in our database for that trait. And if it didn't exhibit that trait, we gave it a 0 in our database. So for each of the trait categories that you just saw, the trophic, reproductive, and habitat traits, we measured functional diversity separately for each HUC-8. So we had 3 functional diversity values within each HUC-8. As I just said, 1 for trophic, 1 for reproductive, and 1 for habitat functional diversity. And once we measured that initial functional diversity value using the functional dispersion metric, the observed functional diversity values were then compared to a null distribution to generate standard effect sizes, or SES values. And these SES values, they standardize functional diversity measurements across HUC-8s. And they're actually similar to z-scores, so they do something similar to what a z-score does in standardizing. And the SES values are determined using an algorithm to shuffle the tips of our trait dendrogram 999 times, and then we measure functional diversity each time, and that gives us a null distribution of functional diversity values, which we can compare our observed functional diversity value to that we measured in each HUC-8. So once the null distribution is determined, we calculate the SES value by subtracting the mean of the null distribution from the observed functional diversity value for a given HUC-8, and then what we do is divide that by the standard deviation of that null distribution. And these SES values allow us to determine if the observed levels of functional diversity in each HUC-8 are greater or less than what we would randomly expect given the species pool. So positive SES values yield a HUC-8 that is more functionally diverse than we would randomly expect, and negative SES values would yield a HUC-8 that is less functionally diverse than we would randomly expect given that null distribution. And also, instead of the raw functional diversity values, these SES or standardized functional diversity values were what we used in correlating with indicators and environmental variables across the SALCC. So to get into our results, just to start off, we first just looked at this correlation matrix, which is depicting Spearman rank correlations between each of our calculations of functional diversity. And because this is a symmetric matrix, we only show the numbers above the diagonal because obviously what's below the diagonal is the same. And here we, excuse me, and here we wanted to know if these 3 different measures of functional diversity were telling us similar or different things. So looking at these correlations here, you can see that trophic and reproductive functional diversity are highly correlated with one another, as you can see by the correlation coefficient of 0.91. But both trophic and reproductive functional diversity are not strongly correlated with habitat functional diversity. You can see the correlation coefficients of 0.48 and 0.36. And these relationships will be depicted spatially in the next slide. So here we're looking at the sort of pretty part of our results. These maps show trophic, reproductive, and habitat functional diversity across the SALCC at the HUC-8 scale. And as you saw in the previous slide, and as you can see here, both trophic and reproductive functional diversity patterns are more similar with one another compared with habitat functional diversity. So I'm just going to start by showing you those two. So on these maps, warm colors indicate high functional diversity, and cool colors indicate low functional diversity. And positive functional diversity values, as I stated earlier, are more functionally diverse than we would expect under random compared to the null distribution. And negative functional diversity values are less functionally diverse than we would expect under random. And it's also important to note that each map is standardized separately based on the regional trophic, reproductive, or habitat functional diversity values estimated. So comparisons between separate maps can't be made directly. And high trophic and reproductive functional diversity is estimated here throughout the Eastern Highland and Appalachian regions. And these are areas of known high fish diversity with large numbers of endemic species. And lower functional diversity is generally seen in coastal plain and Piedmont areas, more so in the trophic map than in the reproductive map. But these are where levels of endemism are lower. And in addition, the coastal plain regions are typically more developed and urbanized compared with the highland and Appalachian regions, which would lend additional support for why we might be seeing lower functional diversity here. So I'm going to switch gears and just show you the habitat map. So looking at this map, you can see that there's really no clear pattern of functional diversity here. There's both high and low functional diversity estimated throughout different huck gates in the region. And I'll touch on why this might be the case later on. So here, this slide is just so you can get an idea of which huck gates had the highest and lowest functional diversity values. And the Altamaha River in southern Georgia had the lowest functional diversity estimates across all three trait categories. For the trophic trait category, the Meherrin River in Virginia and North Carolina had the highest functional diversity estimates. For the reproductive trait category, the lower Ocklaconee River near the Apalachicola had the highest. And for the habitat trait category, the Black River in North Carolina had the highest. And just thinking about some of these measurements and why some might have the highest and some might have the lowest, we speculate that the Altamaha River potentially has the lowest functional diversity because the river's acidity does not exhibit favorable conditions for potential diversity of trophic interactions, reproduction, and habitats. And all three subbasins that portray the highest functional diversity values for each set of traits, so the Meherrin, the Ocklaconee, and the Black River systems, they all have a high degree of undisturbed riparian, so they all have above 90% undisturbed riparian, and they all have close to 0% agriculture and urbanization, all of which would be expected for highly functionally diverse systems. So they're all pretty much relatively undisturbed huck gates, and therefore they haven't been urbanized, and this could explain why we're seeing these higher functional diversity estimates in these systems. So looking at some of our correlations with indicators and some of our correlations with a few other environmental variables, there were no significant correlations between the indicators and functional diversity here, but we're just going to look at some of the loose trends that we see in the trophic functional diversity category. So looking at this table, you can see that trophic functional diversity is positively associated with percent of natural cover near rivers and streams or undisturbed riparian, and it's negatively associated with percent of urbanization and percent of agriculture. And these trends are what we would expect because more natural ecosystems promote more feeding niches, whereas increases in urbanization and agriculture decrease these feeding niches. And we would also expect to see the same trends across the reproductive and habitat trait categories, but that's not what we see here. And again, these associations aren't significant and they're not strong by any means. So we can't extrapolate too much from these relationships at this point. But in order to get at these relationships further, we might need to look at additional models aside from these simple Spearman rank correlations that might kind of pull out or tease out the relationships a bit more. But if we do look at the association between functional diversity and species richness, these were all significant, and we see that the relationships are neither strong nor weak. So this is alluding us to the fact that functional diversity and species richness are not highly correlated with one another, and therefore there's value in looking at the patterns depicted by both of these spatially. So instead of just looking at species richness or just looking at functional diversity, we can look at both to maximize our potential conservation prioritization. So to further demonstrate our functional analysis, we looked at, or we pulled out two specific huck gate or subbasin segments, and these were similar in species richness, but they had different values of functional diversity. So we thought that it would be good to kind of analyze to see why that might be the case. So looking at the lower Savannah River, which is seen here in dark blue, it has 20 species based on our databases, and it's a highly modified river system with numerous large reservoirs and maintained river channels. And switching gears and looking at the middle reach of the Tallapoosa River, which is seen here in teal or lighter blue, has 18 species. It's located in east central Alabama, and it's also altered hydrologically by main stem reservoirs, but it's not extensively channelized, and the subbasin overall is likely less developed than the lower Savannah River. So our database contains two major groups of species for the lower Savannah River. It contains the sunfishes of genera Cynobrytis. It contains the genera Lepomis, Micropterus, and Pomoxis. And it also contains the catfishes of genera Ameiurus and Ixylurus. But for the middle Tallapoosa River, our database contained a much more diverse assemblage, including several endemic species, including two Cyprinella, an endemic catastomid, and several cyprinids and centrarchids. So we estimated lower trophic functional diversity, reproductive functional diversity, and habitat functional diversity for the lower Savannah River compared to the middle Tallapoosa River, which, as I just stated, had a more diverse assemblage of species. So because these two hucks had similar values for indicators already explored in the last slide, so they had similar values for things like percent natural cover and percent ag and urbanization, it's likely that the differences between these two systems are related to other habitat and ecological variables that we haven't explored yet. So as an example, we speculate that the middle Tallapoosa River is not channelized as extensively as the lower Savannah River, and the middle Tallapoosa River subbasin is also much less developed than the lower Savannah River. It potentially has a higher permeable surface, and habitat complexity is likely much higher in the middle Tallapoosa, although both rivers are regulated. And because the lower Savannah River contains primarily catfishes and sunfishes, and there are more shared traits in that system, the functional group for this area is low. And for example, the middle Tallapoosa has more species with a diverse combination of traits, such as Cyprinella gibbsii, which is the only fish species in either subbasin that feeds solely on macrophytes or vascular plants, and also invertebrates and larval fish. And this is important in terms of thinking about things like biotic homogenization, which is the process by which species invasions or extinctions increase the taxonomic or functional similarity within a system. And urbanization and the associated degradation of systems and habitats that come with urbanization has been shown to induce biotic homogenization, which would explain the lower functional diversity in the lower Savannah system compared with the Tallapoosa River. Additionally, the middle Tallapoosa River is part of the mobile river drainage, which is a region of pretty high endemism, while the lower Savannah River is primarily within the coastal plain, which is a region of much lower diversity. And analyzing additional indicators such as channelization and things like permeable surface cover or these endemic hotspots could reveal deeper insight into the differences in functional diversity that were measured between these huck-gates and potentially in looking at a bunch of different individual huck-gates and comparing them with one another. So some overall conclusions and remarks. The revealed regional patterns of functional diversity that we see here are insightful, but when we correlate with indicators at the Huck-8 scale, we don't see any significant results. And this could be attributed to our fish occurrence data. Again, the data comes from many different fish sampling efforts, and ultimately it would be best to use sampling efforts from one systematic sampling program, but this doesn't exist across the entire region. And conducting this analysis at the Huck 12 scale, which is a much finer resolution, might reveal some tighter knit relationships between functional diversity and indicators because of the smaller scale. When analyzing individual Huck-8s, these results, and another thing that's important to note is that when analyzing individual Huck-8s, these results might be most useful, as we did when looking at the lower Savannah and the middle Tallapoosa comparison in the last slide, and also when we analyzed our individual Huck-8s that had the maximum and minimal functional diversity values. It's also important to note that although we mainly focus on functional diversity here, it's really important to consider functional diversity in accordance with other biodiversity measurements, such as things like taxonomic and phylogenetic diversity, to highlight areas of highest conservation priority. So as we're continuing with our SALCC analysis, as more data and indicators become available, we're also working on this same analysis with Mark Scott and colleagues from the South Carolina Department of Natural Resources. And they have standardized fish sampling and environmental data across South Carolina wadable streams between 2006 and 2011. So all of the data comes from one systematic study, whereas one caveat to our fish sampling data from the SALCC is that it comes from many different fish sampling efforts. So it's going to be really interesting to see if the loose trends that we revealed at the landscape scale across the SALCC will be reaffirmed here when we're measuring functional diversity across South Carolina water bodies. And from just running simple Spearman rank correlations, we've already revealed that there are, in fact, significant correlations between functional diversity and environmental variables. And we're currently working on a random forest regression tree model to reveal the most important or influential variables that drive functional diversity across South Carolina. And just right now, from running some of the initial random forest models, some of the most important, or a couple of the most important variables are percent of open water and percent of wetland cover. So I'd like to acknowledge a few people. I'd like to acknowledge some of my committee members, Ben Baser, Bill Pine, Christina Romagosa, and Catherine Phillips. I'd also like to acknowledge Rua Mordecai, Amy Keister, and Amy Benson, Noel Burkhead, and Stephen Walsh. And I'd really like to thank the South Atlantic LCC for the funding for this project. And also, I'd like to acknowledge Mark Scott and the South Carolina Department of Natural Resources for allowing us to work on this follow-up analysis to kind of tie some of these larger landscape patterns with some local data in South Carolina. And at this time, I'll take any questions. Thank you very much for listening. And I have some references if anybody needs, but I'll take questions.
Great, thanks, Josh. Great presentation. We definitely have plenty of time for questions or discussion, so you're welcome to use the chat box or press *6 to unmute.
And these functional diversity layers will be available. Right now, Rua has one of them, and I'm working on the trophic functional diversity layer, and I'm working on sending him the habitat and reproductive layers as well.
Great.
Josh, this is John Faustini. I had a question. Great presentation, by the way. I was struck by how the habitat functional diversity was strongly negatively skewed from that normalization, and I was kind of wondering why you think that might be. Is it just because there's more variability in that, there's more of those habitat metrics, there's a wide range, and that's spread over the whole geography, so when you're calculating that normalized value, you're not likely to get that kind of range of that kind of diversity in a single Huc-8 that you would get across the region? Or is there some other interpretation there?
Yeah, I think that's definitely a possibility. I kind of overlooked that. There were a lot more habitat traits that went into the measurement of functional diversity, which I think definitely could be part of the reason we do see that. Our habitat database, our habitat trait database, also contains some missing traits compared to our reproductive and trophic traits. So I think that could also be part of the reason. I also, I'm not sure. I think it could be the sporadic sampling data. But we do see some decent patterns with the trophic and reproductive maps. So I don't think we could attribute it solely to sampling. I think it has to do with that there are a lot more habitat traits analyzed, and habitat is much more complex compared to our other traits.
Right. It might be interesting to explore, like doing something like if you normalize, if you regionalized it, or like did the coastal plain you just normalize the coastal plain sites within the coastal plain, and for example, because substrate, you're not going to get a lot of gravel and coarse substrates there versus in the Piedmont or the Appalachians.
Right, that's true. Yeah, I think definitely taking into account for future analyses which traits are actually most important, and it definitely comes down to what the region that we're looking at, and I think it's also important to consider what we're trying to get at the functional diversity analysis. Here we were kind of interested in broad patterns, but if we're thinking about specific services or specific ecosystem functioning that we might be interested in for any particular reason, I think it's important to really think about more critically which traits actually go into the functional diversity calculations.
Mm-hmm. Great. Thanks.
Yeah.
This is Mary Davis. I agree with John. Good presentation.
Thank you.
So, yeah, I think it's an interesting approach, and I'm just wondering, what are your thoughts about the scale? Would you choose Huck 8 for a functional assessment if you had better data, or would you go finer? Tell me about scale.
I would go finer, and I think that it's harder to, we might have Huck 12s within a Huck 8 that are very different, and I think being able to see that functionally would be more valuable than this Huck 8 analysis, but I definitely think there's value in both of them. But definitely if the data was available, I would love to do this at the Huck 12 scale.
Okay.
Good, Josh. Just a quick follow-up. This is John again. So, are you able to do that in South Carolina with those data?
Do
the Huck 12 level?
So it's not necessarily. So the data is from stream reaches.
Okay, so you're not going by Hucks at all. You're going at it. Right.
Yeah.
But you will have a finer scale of analysis available then.
Yes, it'll be much finer scale, but no, not by Huck.
Okay. Great.
Any other questions for Josh?
Yeah, actually thinking about using this as an indicator for aquatic integrity, it's interesting that you've got it standardized and the values are greater or lesser than expected diversity. I think the aquatic integrity indices need to be able to indicate is change from desired conditions. And so I'm interested in the timeline of data that you had, 1953 to present or nearly present. Are there enough data that you could show change in diversity, functional diversity in some or all of the regions?
There are definitely, it's broken down by time. We do have the year that the sampling did occur. It's just not, there's some years sampling did not occur. Some years, some of the sampling is not what we would expect based on that area, and we could do it, but I don't know how insightful it would be. I think kind of.
You did it with data like before 1980 and then after 1980.
Yeah, I think.
Yeah,
I think.
like that.
Right. Yeah, I think there might be some value in kind of looking at before and after a specific year, which is a great idea, and it's something we've thought about. And I think it would be even more insightful looking when we do it with the South Carolina data. But definitely, it would be worth doing here as well.
Yeah, it's interesting. Yeah.
Because you're right, it definitely change is what we're thinking about in terms of indicators.
So this is Rua. One interesting thought related to that when we got the sampling data is there may be a way to do some species accumulation curves in that before and after threshold and do some kind of detectability correction for species. There's some pretty well and very fast-running analysis where you can sort of predict out just looking at accumulation using time as a proxy and figure out, like, okay, well, how many species are probably there within some of these traits. So there might be some creative things we could do, either to say, okay, these are just some, some watersheds where we just can't do it, or to sort of predict out, just to kind of make up for an imbalance of some areas being really heavily sampled and others not being heavily sampled. There might be some techniques for detectability correction to try to do that.
Okay, great.
Hey, this is Mark, and another thing that occurs to me is the sort of confounding factor of the Clean Water Act. Some areas will actually be improving, whereas other areas will be getting worse due to land use change and so on.
Right. I definitely think that it's important to consider as well. Yeah, that time breakup might be useful in terms of that as well.
I'm not sure about South Carolina, but much of the Southeast has actually increased in forest cover over that time period as well.
Right. Yeah, I'm really excited to kind of see what the South Carolina data reveals. They have somewhat of 100 environmental variables measured, and obviously a lot of them are, measuring similar things, but there's a lot of kind of potential to reveal what's driving diversity across South Carolina.
Hi, Josh. This is Amy Keister. Thank you for your presentation. Oh, I always forget that. Hi, Josh. This is Amy Keister. Can you hear me? It's okay. I'll skip it.
All right. If there are no other questions, we can move on, but I'll pause just one more time. Don't be shy.
Can you all hear me now?
Yeah.
Oh, sorry. Hi. This is Cindy Bone. I just had a quick question. Josh, I was just wondering what the Altamaha River basin showed, since it's in a relatively, quote-unquote, natural state, and we've been working on a lot of protection in that area, and it's unregulated. Did you see any differences in those hucks?
So, differences in the hucks within the Altamaha River?
Right. Did that would that be something that you could use perhaps as like a reference, since it is a fairly uncontrolled and more pristine river?
Yeah, we did, in that one Huck 8, we did see really low functional diversity, and, we weren't really sure why. We kind of just thought about acidity. But, yeah, it would. I haven't done that, but it would be interesting to kind of look at all of the Huck 8s within the Altamaha. And, yeah, I definitely think that can kind of reveal something valuable.
Okay.
Thanks.
Mm-hmm.
I'm sorry, I need to ask for clarification because the names are so similar. Did you say the Altamaha or the Altamaha?
Altamaha.
Okay.
In Georgia.
And, okay. So was it the Altamaha that had the consistently low functional diversities, or the Altamaha?
It was the Altamaha.
Okay. I was confused. Okay.
Thanks for the clarification, Mary. Yeah, I was talking about the Altamaha.
Okay, yeah, I just mixed those two up. Sorry.
That's no problem.
Yeah, thanks for that.
That's easy to do.
Yeah, the Altamaha is pretty hammered. That's pretty intense ag and with irrigation.
Okay, so that would make sense then why we're seeing low functional diversity.
Right. So did you see I didn't notice when the slide was up. I sort of tried to look in that area, but so on the Altamaha Basin in South Georgia, the lower Altamaha, right after the confluence of the Ocmulgee and the Oconee. That entire whole stretch of the river is unregulated. I believe it's our only unregulated stretch of river in the southeast, at least in the southeast coastal plain. So one would hope you would find a fairly high functional diversity in that river as it's in its more quote-unquote natural state. And I was just wondering if you did, or if that would be a good reference for a reference reach for you all to check whether or not your models are.
So which river system is that?
It's the Altamaha River.
Altamaha.
Yeah, it flows. It's the confluence of the Ocmulgee and I believe the Oconee. Is that right, Mary?
Yeah.
And it flows all the way from that confluence into the Atlantic Ocean. And it should have fairly high, it has very high wetland.
Yeah.
It's pretty low on these maps.
Is it?
Yeah,
look
I mean,
theokeefenokee.
does look low. But yeah, that's definitely something I'm going to look into. It could be.
I mean, because we pretty much most of that river is as protected lands on both sides of the river. It is unregulated, so there's no dams or anything on the whole lower stretch.
But it does have flathead catfish, and has had some concerns over native fish impacts of flatheads there too. So that might be one of the issues.
I'm sorry, I couldn't hear you.
Oh, I just said I was thinking about the flathead catfish introduction issues in some of those rivers too. That I guess we need to go back and take a look at see what the time series of available data for these rivers are. Because any specific river like that, you're right, the Altamaha should have decent data available for it. So we need to come up with a way to graphically present the number of samples that are available for each of those different basins and what the timing of those samples are. So say the Altamaha data are recent data, and maybe the Altamaha diversity has been impacted by flathead catfish, then it might not have reflected what the pre-flathead catfish introductions were like in that community, even if the riparian areas are relatively intact.
Yeah, but I don't think the Altamaha is experiencing that kind of an influence. You just may want to check. And, I know that I haven't been involved in this for a while, but I know the Marist data didn't include a lot of threatened endangered species or at-risk species data in it, by design. So could just mean that you need a little, it's the age-old problem of not having the data that you really want. But that might be something that you want to take a look at, because if you would, the Altamaha is kind of our standard in the southeast for restoration. For protection, and so it is our reference river for restoring flows and different things like that. So it has a fairly intact faunal.
Okay.
So that's my understanding, but I'm not an expert on it. So
yeah, you're right.
You might want to check with some folks.
It's kind of interesting looking at the drainages out of the Theokeefenokee. The Theokeefenokee doesn't rate well on either of these indices, and the St. Mary's doesn't rate well. It usually rates about the same as the Theokeefenokee, but then the Upper Swanee rates pretty highly. And the Upper Swanee is just about the most acidic river in the region. So I don't know. I think the change in diversity is what's more meaningful here for an aquatic integrity index myself. And I know it's hard to get. I'm not saying that we can get it, but there's just a lot at play in all of these.
Okay, yeah, well, I'm definitely going to try and consider how we can incorporate that change and if it's possible.
Yeah, great discussion.
Yeah, thanks for all the feedback.
We might have time for one more quick question if someone wants to sneak one in. All right. Well, hearing then, I'm going to wrap up with some updates from the South Atlantic LCC here before we log off at 11. Let me put my slides back up here. So I just wanted to highlight that we have a redesigned website and new Facebook page, and we're going to get a quick update from RUA on indicator revisions in preparation for Blueprint 2.1. So if you haven't visited our website in the new year, you'll find that it has a very new look. It's at the same URL, southatlanticlcc.org. But it has a much more modern look and feel. It should be much easier to find key information about who we are as a cooperative, what we do, and how we do it. So the fundamentals are still there. You can still post blogs and events to share with the rest of the community. But just be aware that when you log into your account on the new website for the first time, which most people probably don't need to do consistently, but if you ever do want to share a blog or an event, you'll need to sign into your account. And the first time you do that on the new site, you're going to need to reset your password. So we could import all the account information, but we couldn't import passwords to maintain the security of everyone's profile. But regardless, password or not, you're still going to get the newsletter if you have an account on the South Atlantic website. And we're also now on Facebook, so I encourage you to check out South Atlantic LCC Facebook page, and you can link to it from the footer on the website. And now Rua is going to give us an update on the status of Blueprint 2.1 indicator revisions.
Sure, thanks, Hillary. So updates were basically waterscape and freshwater aquatic indicator revisions are done at this point. We're doing some cleaning up and formatting and getting things ready for use in Blueprint 2.1. Almost done on the marine side. Still got a few more things to wrap up on the marine indicator sides. And now starting to move on to develop Blueprint 2.1, so shooting for summer this year. And so we're starting to get some of the models going and set up some pieces. So expect from folks on the phone, I expect we'll have some stuff for you to review in a few months to start taking a look at some interim products.
Great, thanks, Rua. So previewing the next Third Thursday Web Forum, if you are intrigued by Rua's sneak peek of progress toward Blueprint 2.1, you should definitely tune in February. Our next Web Forum will be on February 18th, same time, same place, right here at 10 a.m. And Rua is going to be talking about improving the Blueprint updates and discussion on Blueprint 2.1 and indicator revision. So we're continuing with that yearly revision cycle for the Blueprint and the underlying indicators. So as Rua said, this is a smaller data-driven update with similar methods as Blueprint 2, just improved indicators for freshwater aquatic, pine and prairie, and the marine systems. So we'll leave plenty of time on that web forum for discussion and feedback on the results of indicator testing and the next steps toward developing 2.1. And finally, I always want to make sure folks know how they can get involved in their cooperative. So in addition to contacting staff or someone else involved in the cooperative, you can always check out the frequent updates and events that we post on the website. It'll help you learn about upcoming projects, opportunity to serve on teams, or how to access spatial data and the Blueprint. So definitely would encourage you to join our web community, get access to our monthly newsletter. And with that, I'll just open the floor for any additional questions that anyone might have before we sign off, either about Josh's talk or what's going on in your LCC. All right, well, that's it. Thank you so much, Josh, for your great presentation. We appreciate you all attending. Hope to see you next month.
Thanks a lot.
Enjoy it.